Hello students, today we will continue the chapter Sexual Reproduction in Flowering Plant. Topic we are going to discuss is pollination. In our previous class, you have learned that the male and female gametes in flowering plants are produced in the pollen grains and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve the objective. Transfer of pollen grains to the stigma of a pistil is termed pollination. Depending on the source of pollen, pollination can be divided into three types: autogamy, diatonogamy, and genogamy. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grain from the anther to the stigma of the same flower is termed as autogamy. Seen in plants which produce chasmogamous and cleistogamous flower. Chasmogamous flower have exposed anther and stigma. Cleistogamous flower do not open at all. In this type of pollination, the pollen grains are transferred from the anther to the stigma of a different flower but of the same plant. Genogamy. It is the transfer of pollen grain from the anther to the stigma of a different plant. Agents of pollination. The process of cross-pollination requires the help of biotic and abiotic agents like animals, birds, wind, insect, water, and other agents as pollinator. Pollination by wind is also known as anemophily. There are only a few flowers that use wind pollination, as their features are greenish, small, and odorless flower. The male part of the anemophilous flowers tend to produce very large quantities of pollen, and the stigma, the female reproductive part of a flower, are very large, sticky, and feathery to extend completely outside the flower. Thus, the pollen is more likely to reach them. Coconut, plum, maize, grasses, and all gymnosperms are the best examples of wind-pollinated plant. Pollination by insect, entomophily. Insect-pollinated flowers are large, colorful, fragrant, and rich in nectar. Example: mango, rose. Pollination by water, hydrophily. It is very rare. Pollen grains of many such species are long, ribbon-like, and are carried passively inside the water. In some species, pollen grains are protected from wetting by a mucilaginous covering. Example: Hydrilla vallisneria. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetical mechanism and prevents self-pollen from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flower. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female. This condition prevents both autogamy and diatonogamy. Pollen-pistil interaction. Pollination is just the beginning, or rather the first step of pollen-pistil interaction. Pollination does not guarantee transfer of right type of pollen. If pollen is of the right type, the pistil accepts the pollen and promotes post-fertilization events that lead to fertilization. The pollen-pistil interaction begins with pollination followed by pollen adhesion to the stigma. The pollen tube penetrates through the stigma and the tube grows through the style and reaches the ovary. Once it reaches the ovary, the tube penetrates it and reaches the micropyle of the ovule and enter into the embryo sac. Here the two male nuclei fuse with the megaspore and the vegetative nucleus to form the diploid zygote and the triploid endosperm respectively. This fusion of male and female gametes is known as fertilization. Artificial hybridization is one of the major approaches of crop development program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and stigma is protected from contaminations. This is achieved by emasculation and bagging technique. Emasculation. If the female parent bear bisexual flowers, removal of anther from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step of removal of anther using forceps is termed as emasculation. In the case of unisexual flower, this step is not necessary. Bagging. Bagging is the protection of emasculated flower from contamination by undesirable pollen grain. Here the flower is marked by a bag. Still, the flower attains receptivity. In unisexual flower, bagging is done before the flowers are open. Emasculation and bagging ensure that female flower is completely protected from contamination. Hope you have enjoyed the presentation. Thank you.